Hi, my name is Lars Maegdefessel. I'm this year's Vice Chair for the ATVD Vascular Discovery meeting, and I'm joined today by Stefanie Dimmeler from the Institute of Cardiovascular Regeneration in Frankfurt. Stefanie will give the keynote lecture this year about RNA biology in vascular diseases. Maybe for the people who could not join us today, maybe you could introduce us a little bit to what you'll be talking about in the session.
So actually I will talk about mechanisms of vessel growth, of course, so how blood vessels grow, particularly after ischemia. In the heart you need this for repair and regeneration potentially, also in other organs. And we will focus on non-coding RNAs.
Yeah, so non-coding RNAs is a pretty big species or mechanism. So I know you've been really focused on microRNAs for a long time and now have kind of expanded into other forms of non-coding RNAs. So what's your take on the different subspecies of non-coding RNAs?
So of course microRNAs are, as you said, the well-established ones, and also there's, I think, some progress towards also clinical studies. So I will talk in my first part of the presentation of a microRNA named microRNA92A, where we have developed inhibitors which are now in phase 1, so are currently tested for safety in humans. And we aim to use these inhibitors in patients with acute coronary syndromes because these antimers have anti-atherosclerotic and endothelial protective effects, but potentially also in chronic ischemia to repair or maybe pertain endothelial cell function and microcirculatory function in this context.
Yeah, very exciting. So I mean, as you mentioned, you've been at the very forefront of using stem cells and pushing that into clinical trials. Now you're again at the very forefront when it comes to non-coding RNA-based therapeutics, which is really exciting, I think. So where do you see maybe pitfalls or problems that could arise going from basic science into clinical?
Yeah, as always, only few things will finally made it and be then successful. Also for cell therapy, we are still not clear whether it works or not. So phase 3 clinical trials are running, but it's a long time. But if you don't do it, you never will know whether it works or doesn't work. I think with microRNAs, I'm quite confident because you can use small antisense molecules for inhibition. I think for long non-coding RNAs or circular RNAs, these are the other species of non-coding RNAs. They are very exciting, but we know even less about their biology. And although maybe it's more difficult to inhibit them because you have to use other types of oligonucleotides like gapmers, which are not that well established. And overexpression always is difficult, as we know from gene therapy in the cardiovascular field. Targeting endothelial cells or cardiomyocytes is not that trivial in clinical settings.
So you mentioned the first part will be on microRNAs. So the second part, I assume, will be on long non-coding RNAs, which I think for many people who've been working with microRNAs and have now transitioned into the long non-coding RNAs, it's not as trivial as or as easy, I guess, as doing microRNA-based research. So what's your take on this? How do you?
So surely I'm not only believing, but there's clear evidence that several long non-coding RNAs have important function in the vascular system. So we have first genetic mouse models. And also in clinical settings, we see associations of the expression patterns of long non-coding RNAs with disease progression, like in atherosclerotic disease, where we see that MALAT1 expression determines prognosis in plaques. But of course, it's a very, very young field. And I think while we can understand maybe the biology, meaning the effects of a knockout, the biological mechanisms underlying these effects are really complex. So long non-coding RNAs can have multiple effects. They can work epigenetically, they work as potentially microRNA sponge, but also more importantly, they scavenge RNAs, they interact with proteins. So it's difficult to do the biochemistry and the biology correctly also because it's technical demanding to gain mechanistic insight.
Yeah, very nice. So we've already talked a little bit about therapeutic potential. One other field where people have been exploring non-coding RNAs is more the biomarker field. So what do you think? How could maybe non-coding RNAs fill gaps in this area?
So I think the microRNAs, again, because they are more longer known and also better established, there are clear evidence that they are somehow correlating to disease settings. But also here we need technical developments to really measure them reliably. So I think at the time being we still have not yet the setup where we can measure them in a way like we have ELISAs for proteins. And I think there needs to be a technical development. With respect to long non-coding RNAs, there are a few exciting studies out, but also it's more difficult because these are long non-coding RNAs, so they are longer and they are usually nuclear, so they have to be exported from the nucleus to the cytoplasm, to the, let's say, extracellular space. So maybe it's not that trivial to detect them as for microRNAs, which are maybe smaller and can be easily released.
Yeah, maybe one last question. So this year's meeting was really focused on, or the theme was from genes to medicine. So precision medicine is one big theme, I guess, in biological, but also biomedical research. So what's your take on precision medicine? How do you see potential implications with the work that you are maybe doing more on the basic or translational level and its potential transition into the clinical?
So I think precision medicine, of course, is the goal to go in the future, because we need to have a tailored treatment for patient subpopulations. How and if long-coding RNAs play into this, of course, it's not a trivial question. But I believe because a lot of these long-coding RNAs are human-specific, we may use them to better understand the pathophysiology in humans, and maybe they also more distinguish between biological settings in humans. So I have a hope that we may use this as a tool, at least in first single-cell sequencing studies, where we try to explore the transcriptome in individual cells. We see really clear clusters of long-coding RNA expressing cells, and this might be an attempt so that we understand better the expression patterns and maybe find very interesting populations of immune-competent cells, which may change atherosclerosis or also may change the prognosis after acute myocardial infarction. But this is, I would understand, maybe easier it would be to have imaging tools or things like this in the first glance.
So thank you very much, Dr. Dimmeler, for this very nice interview. We're really looking forward to the presentation.
Thank you very much.